Hello and welcome to another edition of Zog Science. Today we're going to be talking about enzymes and how they function and how they are extremely important to the function of our bodies. So enzymes are catalysts that speed up chemical reactions within organisms, and they're incredibly important because if they don't speed up those chemical reactions, we would actually not be here today. We've talked briefly about cellular respiration and photosynthesis, and both of those are chemical reactions. And if they were to just occur at their normal, natural rates in our bodies, obviously photosynthesis wouldn't, but cellular respiration for us, we would not have enough energy in order to live, in order to survive. So it's very important that we have enzymes. So what exactly is a chemical reaction? Okay, it's the process of changing one set of chemicals, which we call the reactants, into another set of chemicals, which we call the products. And the way we do that is we rearrange the atoms. Okay, there's the law in the universe called the law of conservation of matter, which is that matter can never be created nor destroyed, but it can be the form of it can be changed. So what's going to happen is that the bonds that are holding the reactants together are going to be broken, which requires energy. It takes energy in order to break those bonds, and then new ones are going to be formed in the products. So for example, in this reaction, the bond holding the hydrogens together and the bond holding the oxygens together has to be broken in order for them to be rearranged into our water molecules. So what exactly is a catalyst? Okay, it's a substance that speeds up the rate of a chemical reaction by lowering the activation energy. Okay, and the activation energy is that energy that it takes in order to break the initial bonds of the molecules. And so it's basically is the energy that you need in order to start a chemical reaction. Okay, so there's two different types of reactions. There's energy absorbing reactions and energy releasing reactions. And both of them still require activation energy in order to get started. We kind of call it like an energy hill, where you require, again, that certain amount of energy in order to kind of get over the hill and get the ball rolling to get from your reactants to your products. So that's our activation energy right here and right here. Again, it takes energy in order to get the reaction going. So this would be, over time, you require this kind of energy. So enzymes speed up chemical reactions by lowering that activation energy. So, without an enzyme, you might have this amount of energy that is required in order to get the reaction going. But then with the enzyme, you only need this much. So therefore, your reaction can occur much more quickly because you require less energy. So, what are enzymes? They're proteins, okay, and we already said that proteins are made up of amino acids, okay, so they are a polymer. They're one of our macromolecules, and they're biological catalysts, right? And they're used by cells to trigger and control chemical reactions. And without enzymes, several reaction cells, like cellular respiration and photosynthesis, would never occur or happen too slowly to be useful. Again, because you wouldn't have the energy in order to get them to occur in the first place, or it just would take too long for that to happen. All right, what is a substrate? Okay, substrates are the molecules that the enzyme binds with. And we just have that specific name for them so we can distinguish them. All right, and the substrates are the reactants that are going to undergo a reaction that is going to be catalyzed by the enzyme. So it can be either a single molecule that's going to be broken down, or it can be several molecules that are going to be put together in order to form your products. And one of the key points is that enzymes are very specific and are only going to work with certain substrates. And the reason why they only work with certain substrates is because of the shape of the enzyme and the shape of the substrate. So the way that we call this is we call this the lock and key kind of theory or hypothesis. So basically what happens is that you have an enzyme, okay, and the site on the enzyme where the substrates bind is called the active site, right? And they bind to one another based on their complementary shapes. So one substrate can only bind with one enzyme, just like a lock has a specific key that is going to be able to unlock it. So we call this enzymes being very specific, in that they only work with one type of substrate. So if you just because you get excess amounts of a particular enzyme, it doesn't mean that you're all of a sudden going to have your whole body going through reactions, because it's only going to work with a specific substrate or a specific reactant to then produce that product. And it's all based on the shape of your enzyme. And while we're talking about the shape of the enzymes, I want to show you something that is very cool. So there's this very cool website called the the National Center for Biotechnology Information, and this website basically compiles a bunch of the research data that is done in the United States, so that everybody can share it with one another. And one of the things you can do is you can search for the structure of particular enzymes, and you can actually see what their shape is, and then based on the shape you can predict what they are going to be able to bind with. For example, we talked in class a little bit about amylase. Okay, remember we said that amylase was an enzyme in your saliva that helps you to break down food as you are chewing. So what we can do is they actually can create models of the protein, so that we can look at it and see it. I'll explain what this is in just a moment. Okay, so here is our three-dimensional structure. So one of the things we kind of forget sometimes is that molecules have a three-dimensional structure, and proteins in particular have a three-dimensional structure. Even though they're a long chain, that chain actually folds on itself in order to produce these three-dimensional structures. So down here at the bottom, there's actually two pieces to amylase, and those pieces are bound together, but those two separate pieces, they have a chain of amino acids. So each of these letters represents a particular amino acid, and this is the specific chain, and it's a little different, but that's essentially the same, and that's what's down here. If we look up here, what I can do is I can actually drag my three-dimensional structure around, and I can see specific spots of where things are going to bind. So these circles right here, they sort of show the active sites in where the substrate is going to be binding with the amylase. Okay, and I can do this not only for enzymes, but I can do this for any other protein as well. So if I go back, I'm going to look up hemoglobin. Right, hemoglobin is a very important protein, and it's what carries. Here's the human version. It's what carries the oxygen in your blood. So again, I just pull this up, get it nice and big. Okay, and what we can see is that when we do this is that we've got four distinct regions. Again, they're all very similar to one another. I can zoom in and take a closer look, right? And I can actually see that there's kind of four spots, right? Four spots where my oxygen is going to be binding. So each hemoglobin molecule has four spots and can hold four oxygen molecules. Okay, and there's different ways that I can view it. I believe that there is a way that you can actually see the okay, so here's kind of those active sites. You can kind of get rid of things. That's pretty cool. I like messing with all this kind of stuff. But yeah, it's pretty powerful stuff. And we'll mess with the NCDBI website, a couple different times throughout the year. Okay. Now, this three-dimensional structure is very important because, again, enzymes are specific, so they have to bind with a specific substrate. So issues can occur, okay, if things are changed. We'll get that to a moment. But enzymes are not affected by the reaction. So whenever they go through a reaction, they are not changed, and therefore they are reusable, which is another very important characteristic of enzymes. So you can put a very small amount of enzyme into your body, and it will carry out all the reactions that it possibly can until there's no more substrate left, which is great because that means your body doesn't have to spend a lot of energy producing your enzymes constantly. As long as it can make a little bit, those are going to be able to be reused. Yeah, enzymes can change shape slightly to fit your substrate a little bit better. So they kind of, instead of it being exactly hard and fast like the lock and key, they can actually move just a little bit. They change their shape. Again, they still only work with a specific enzyme, but they just, that three-dimensional structure that we saw, it actually bends just a little bit. Now, enzymes, like I mentioned before, can be affected because they have this highly specific three-dimensional structure that is very important in terms of the specificity. That there are factors that can affect that shape, and those factors are temperature and pH. Okay. In the last set of notes you did, we talked about the fact that your body maintains a specific pH for the enzymes, and that's exactly the reason is because the pH of your blood or your stomach acid affects the shape of the structure. So if we were to go back and take a look here, okay, the different structures here, the different amino acids here, if you have more hydronium ions or more hydroxide ions, those ions can actually pull and change the interactions of the different amino acids with themselves that cause them to fold, which therefore changes the shape of my enzyme. Okay? Same thing with temperature. Different temperatures, it affects the way that the molecules interact with one another, and it changes the shape. So when this happens, this is called denaturation. Okay, so the enzyme is denaturing, and again, this is the reason why fevers are bad. Okay, this is why you don't want to have a fever. The fever is killing maybe the bacteria that is trying to infect you, but the problem is that all of your enzymes and proteins in your body only operate within a specific range. So if you get too high of a fever, even though you're helping your body fight the disease, you're actually also killing yourself, or you're harming yourself, because your proteins can't handle that temperature. So the enzymes in our body work best at about 98.6 degrees Fahrenheit, 37 degrees Celsius, and a pH between 6.5 and 7.5. Obviously, your stomach acid and the enzymes there work best at a much lower pH, around 2, but for the majority of your body, it's going to be very close to neutral. And just kind of giving an example of an enzyme that breaks down hydrogen peroxide. It's called, catalase. And an important feature of enzymes is that when they're named, they almost always have this ASE ending. So whenever you're looking at reading something and you see that ASE ending, that's a very large hint. That it's not. That it is a enzyme. And so an example would be if you were to put H2O2, hydrogen peroxide, catalase is in hydrogen peroxide. Catalase is in potatoes. So if you put hydrogen peroxide on the potato, it's going to start bubbling and giving off oxygen and hydrogen gas. Giving off oxygen gas, and then there's water left over. That's going to be because it's raw. It hasn't been denatured. But if you boil a potato, then what happens is that it denatures the enzyme, and then your potatoes is not going to cause the bubbling. All right. So I believe that is everything. One last thing. Enzymes can actually be turned on and off. So if there are specific binding sites on the enzymes that can be turned on and off through another chemical, and that allows your cells to be able to turn off certain enzymes during stages of mitosis or during intense exercise or something like that. All right. That's all for today, and I will see you next time.